Good morning, students. Welcome to our class Biological Science. Dear students, in the previous class we have started our lesson plasma membrane. In the previous two classes we discussed about plasma membrane. What is plasma membrane? How it involves in the entry or exit of materials or the substances through it. Yes or no? The grape activity or Kishmis activity. Okay, we noticed that one, or in the potato cube activity, how the water enters into the substances, how it leaves the substances through this plasma membrane. Do you understand that? And also we all are aware to how to prepare the saturated sugar solution, saturated salt solutions. Okay, how to make this, all these things we have an idea about it. In this section, we are going to discuss about the characteristics of this plasma membrane. The first one is the plasma membrane is impermeable. Sometimes it is. So, if the plasma membrane, if impermeable means if the membrane does not allow the substances to pass through it. Okay, it's not allow the substances to pass through it. In that time it is impermeable. In the second case, permeable. Permeable membrane. In the case it allows the both solvent and solute to pass through it. It is permeable. If it is not allows the solvent and solute to pass through it is impermeable. And the next one is semi-permeable. If the membrane allows the passing of solvent and not allow the solute, that is semi-permeable. Okay, fourth one is selectively permeable. If the membrane allows the solvent and only selected solutes to pass through it, that is selectively permeable. Do you understand that? So in this way, it is called as in this way, impermeable. If the solvent, if the membrane does not allow the movement of any substance, either solvent or solute, we know very well, solvent means and solute means. What dissolve in the solvent is solute, which can dissolve the solute is solvent. Okay now? Permeable. If the solvent and solute both are pass through this membrane is permeable. If the solvent is pass through the membrane and not the solute is semi-permeable. If the membrane allows the solvent and only some selected solutes, it's called selectively permeable. Okay, so dear students, we have already discussed so many experiments about the osmosis. What is osmosis? The movement of materials from low concentration to the high concentration. The movement of materials from low concentration to high concentration. In this section, we are going to discuss that one more experiment about this plasma membrane. Okay now, how the materials are passed in the animals, how the water is moves. Okay now, the movement of water in the animals. So, I am going to explain one experiment. This experiment will help to us how the movement of water takes place in the organisms. Okay, so movement of water in the animals, apparatus we required. Other apparatus we required 2 rags, teaspoon, okay, jars, beakers, water, diluted hydrochloric acid or toilet cleaning acid, measuring tape, long strip of paper. Strip of paper. Strip of paper for measuring. So dear students, these are the materials we required. One is 2 rags, base load, 2 teaspoon, beakers, water, diluted hydrochloric acid or tincture, toilet cleaner, strip of paper for measuring. Okay, next one, procedure. The first point is take 2 raw eggs. Take 2 raw eggs and keep it in hydrochloric acid. The students, equal size raw eggs. These are equal size raw eggs. Size equal ga undali. Okay na? Take the raw eggs. That is the first point. Take the 2 raw eggs which are in equal size and keep it in hydrochloric acid for about 4 to 5 hours. What will happen? Think once about it. A hard shell, egg shell, which is made up of calcium carbonate, it dissolves. కోడిగుడ్డు పైప్ ఉంటుంది కదా, అది కరిగిపోతుంది. Okay? The egg shell which is made up of calcium carbonate, it is dissolves. After 4 to 5 hours, take the eggs outside. Take the eggs outside and wash it under the tap water. Okay? The first one is keep the raw eggs in hydrochloric acid for 4 to 5 hours. After that, what happens? The calcium carbonate egg shell is dissolves. So, then by using that tablespoon, take these 2 eggs outside and wash the eggs under the tap water. Okay na? And the third point, measure the circumstance of the each egg. Okay, measure the circumstance of each egg at the widest place of the egg with the help of this strip of the paper, long strip of paper at the widest place. You understand that? Measure the circumstance of each egg with the help of the strip of long paper at the widest place and mark the place with a pen or pencil on the paper. Do you understand this? By taking these 2 eggs from the hydrochloric acid with the help of the tablespoon and wipe it, wash it or wipe it under the tap water, wash it and wipe it and measure the circumstance of each egg by using the strip of the long paper or long strip of the paper at the widest place and mark the place where you measured. Okay na? So mention the size of the wide of the egg. Next, prepare saturated sugar solution. Okay na? Take 100 ml of saturated sugar solution in a beaker and take another 100 ml of tap water. This one tap water, this one saturated sugar solution, and place the 2 eggs in these 2 beakers. Take 100 ml of saturated sugar solution in 1 beaker and 100 ml of water in another tap water in another beaker and keep these both eggs and leave it for overnight. Leave it for overnight. In the morning, observe what has happened. In this case, the size of the egg shrinks. In this case, the size of the egg is increases. What's the reason behind it? In the case or in the size of the egg which is present in the saturated solution or salt water solution, whatever it is. Salt water solution also you can take it. Salt water or saturated sugar solution, whatever it is. So in the case of salt solution, okay, where the egg is present, it shrinks in its level. The egg is shrinking in its size. In the case of egg that present in the tap water, it has increases in its size. What is the reason behind it? Dear students, there is a explanation about the shrinking of size in the egg and increasing of size in the egg that present in the tap water. Because by the process of osmosis, the water contents that present in the egg from less concentration to enter into the high concentration. In this case of tap water beaker egg, in this case water enters into the egg because of it increases in its size. Okay, so because of the osmosis, water always moves from low concentration to high concentration. Here, if the water moves into the moves into the egg. Here, the water comes out of the egg. That means if the water passes or if the substances passes out of the cell is called exosmosis. What is this? Exosmosis. Okay now, if the materials comes out of the cell is exosmosis. If the materials entered into the cell is called endosmosis. Do you understand this? So here we perform this one activity. Take two equal sized eggs and keep it in hydrochloric acid or toilet cleaner for 4 to 5 hours. What has happened? The calcium carbonate shell, egg shell is dissolved in its level on the egg. So take the 2 eggs outside after the 5 hours and wash it under the tap water and measure the circumference of the egg with the help of a strip of the long paper or long strip paper at its widest. Next, and mark the wide of the egg. Now keep the one egg in the concentrated salt solution and another egg in the tap water solution. Both contains 100 ml of solutions. Okay, now water and salt water. So in the case of egg which is present in the salt water, it's shrinking in its size because of exosmosis. That means the materials or the substances comes out of the cell. In the case of the egg which is present in the tap water, it swells in its size or increases in its size because of the end osmosis. Okay, so that is the reason. So dear students, in this way the materials are moved in and out of the animal's body. In the next section, we will see how the plasma membrane works. Another 2 experiments are there. I will explain about it. Okay? Thank you. Thank you very much.